Alright, so we're going to talk about DNA replication, transcription, and translation. So, the first thing we're going to look at is DNA replication. This is going to be, if you remember, in the S phase of interphase, we had to replicate or duplicate our DNA in order to have 2 copies of each of them so that when we split the cell in half, we had enough. So, the first thing we're going to look at, this is what we call DNA replication. So this is happening in the nucleus. And what's going on here is that you have these two, this strand and this strand, are being unwound. And this unwinding is done by an enzyme called helicase. Alright, so helicase is an enzyme that unwinds the DNA. If you remember that all of these DNA strands are held together between the bases, right in between here, by hydrogen bonds. Hydrogen bonds aren't the strongest in the world, so that this enzyme can easily break apart the hydrogen bonds so that we have one side of the DNA strand and the other, so that we can make copies of it. So if you look here, helicase was our one enzyme that was unwinding it. And then we have what's called DNA polymerase, which is another enzyme. If you notice, any enzyme ends in ASE. DNA polymerase is what actually helps copy the DNA. So if you look at this enzyme here, we have all of our little nucleotides, which our nucleotides are our building blocks for DNA. So we're going to have each of these, which obviously is made of a phosphate group, a sugar, and then a base. And then these are going to be inserted in their matching pairs, so A with T and C with G, by DNA polymerase. So basically DNA polymerase works on this side and it works on this side. Don't worry about the fact that it goes in opposite directions. It does, but we're not worried about that. But this is how we get our 2 new sets of DNA. So we've taken one strand of DNA, and now we have another, and we're making another here. Okay, so that's how we get our DNA replication. So we have helicase, polymerase, and we replicate in S phase of mitosis and meiosis. Right? So now we've talked about that. We talked about DNA a lot. What now we're going to look at is something called RNA. So this, we had deoxyribonucleic acid for DNA. This is ribonucleic acid. All right, so those are down there. Ribonucleic acid is single-stranded. All right, so we don't have a double strand like we had in our DNA. We just have this one single strand of bases and the backbone. All right. The sugar in here is not deoxyribose, it's ribose. So that's our sugar. So that's another difference between DNA and RNA. And the one big thing between DNA and RNA is that, remember, we had C, G, A, and T in DNA. So thymine was our T. In RNA, we don't have thymine. We actually have something called uracil. So this is represented by a U. All right. So instead of A, C, T, and G, we have A, C, G, whoops, wrong thing, and U. All right, so A, C, T, and G over here, U. Okay, so those are our differences between DNA and RNA. In RNA, the U is going to match up with A. So it basically just takes the place of T. It acts the same, pairs up with the same things. You're just having a U instead of T. So if you have two things written out, you'll be able to tell if it's DNA or RNA by looking at if it has thymine or uracil. So that'll be one big clue when we're going through all this. All right, so when we have RNA, we actually have 3 different types of RNA. All right, we have something called messenger RNA, which is mRNA. And this is going to be what actually goes into the nucleus, copies itself from our DNA strands, and then comes back out of the nucleus. All right, so this is our messenger. It does what it says. All right, ribosomal RNA. If you remember, we have ribosomes. Ribosomes actually are made up of RNA. So we have something called rRNA, which is going to be our ribosome. So when our ribosomes come in and help with our protein synthesis, this is the form of RNA called rRNA. And then our last one is called tRNA. And this is a transfer RNA. And basically, this is going to match up, we'll show you later, it's going to match up with our mRNA, and it transfers this amino acid. Okay, so it transfers amino acids so that we can make a protein. So if you remember, amino acids are the building blocks of proteins, so we have to use those and put them all together to actually make our protein. So transfer RNA is going to be the RNA that does that. Okay, so transcription and translation. We had our replication. Now this time we have two different things that happen. So we know that DNA holds all of our genetic information for our cells. So this is how you look, how many freckles you have, what color hair you have, how tall you are, how short you are, etc. And so in order for the DNA to actually have all of that happen, so if you have really, really pale skin or really, really dark skin, all of that is shown through proteins that the cell makes. So we have to have a way to get from our DNA and to make a protein from that, because this is our genetic information that's telling the cell, hey, you need to be doing a lot of dark colors for the skin or a very, very light color. All right, so this is our process on how we're going to do it. So, for example, I have red hair. So all of my hair cells on my head need to have a red pigment, which is going to be produced by a protein, and it will be presented by my cells. So my DNA inside my nucleus of all my hair cells have that information for my red pigment, unlike most other people in the world. All right, so my cells need to get my information from my DNA in the nucleus back out into the cytoplasm to make those proteins so the cell can make the red pigment. And this is how we're going to do this. Okay, so DNA is only in the nucleus. How do we now figure out how to move information? All right, if we can't get DNA out of the nucleus because it is too big. So it is not allowed to leave this area. But all of our proteins, if you remember, all of our ribosomes are out in the cytoplasm. So we got to figure out a way to transfer information that way. So this is where this all comes in. So transcription is going to be something that happens in the nucleus. So we're going to use our RNA to take genetic messages from the DNA out into the cytoplasm. So if you remember, of those three types of RNA I showed you earlier, mRNA, which is our messenger, is going to be the one that's in the nucleus making a transcription or transcribing your DNA. And this little guy, because he's smaller and he's one strand, can get out of the nucleus, unlike our DNA. And this is a lot shorter. If you remember, our DNA all wrapped up into a chromosome was really, really long. RNA is only going to take, for me example, this is only going to get that little information for my red pigment for my hair. All right? And then transcription happens in the nucleus, and then translation is going to be what's happening in the cytoplasm. Okay, so transcription is the nucleus. Basically what happens, if you remember in DNA replication, we opened up the DNA strand, so we unwound it, and we opened up the two sides. And remember, that was a helicase enzyme. That was unwinding, so that's still there. It's not shown in this picture, but it's still there. And then this time, because we're not making DNA, we are making mRNA. We use an RNA polymerase instead of a DNA polymerase. So same thing, it's an enzyme to make the polymer of RNA. All right, so you're adding all those nucleotides, making the polymer, you get RNA. So basically, you're doing your same exact thing, the pairing with DNA, or the bases, just like we did with DNA. However, if you look here, we have an A and a U matched together because remember, we don't have T in RNA. So anytime you see an A on the DNA strand, then you're going to match it with a U. So here, 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 and here. All right, so other than that, same exact thing. We're going to now make, let's say if this is the gene for red hair, I'm going to make this whole mRNA strand, and then it's going to go into the cytoplasm. So I want you guys to pause this real quick and make your complementary mRNA strand to this DNA strand. So make whatever pairs you're going to have from that. All right, so we had transcription in the nucleus to get our mRNA. The mRNA now moves out into the cytoplasm where we're going to have translation. Okay, so what happens is, if you remember, our ribosomes are our ways we make proteins. So this is our rRNA. Our messenger RNA is going to pair up with a ribosome, and the ribosome is going to help make our protein. All right, so our mRNA comes in, and what happens is our little ribosome is going to read our mRNA strands in a set of 3 bases. Okay, so it's going to read 3 at a time. It's not going to read each single one like we were doing when we were replicating it or transcribing it. This time it reads it in sets of 3, which is called a codon. So it's basically a code to get the correct amino acid that you need for your protein. So what's going to happen is when it reads a set of 3, the codon, a tRNA is going to come in that has the complementary pairing, so A with U, C with G, and U with A. All right, that's going to come in with an amino acid attached to it and come in and pair up with the mRNA. And this little part right here that's the complementary side, this is called an anticodon. So that just carries the information of the correct amino acid we need for our mRNA strand. Okay, so basically we've had our mRNA, it's taken the copy of the DNA, and now we're making the red hair protein for me, example. And we're putting all of these together and bonding them together to make a growing protein. Right, and they're linked via a peptide bond, which basically just means a protein bond. All right, so when you go to do this, what happens is when we had the codes, those 3 sets, those codons, you're going to look at it and use this. So we've got all of these amino acids, and we've, don't worry about, just, you'll always have this, so the names are a little confusing and a little longer, but that's okay. The one big important thing I want you to know is the start codon. So anytime you have a protein or an mRNA strand, you're always going to have a part on it that has a codon that says AUG, and that's always where the protein will start. So my red hair protein started with an AUG, which codes for a methionine. So if you look on this, if you see AUG at the start of your mRNA strand, you're going to see A, and then you go to the second one, which is U, and then G. So AUG gives us a methionine, which is also called our start codon. You do need to remember this. This is important. Okay? So we only have 1 start codon, but we have 3 stop codons. So if you look, right here says stop, and right here says stop. I'll highlight this for you. Right there. Okay, so these are UGA is a stop codon. And we also have 2 right here. So we have UAA and UAG. All right. You'll always have this wheel. So the stop codons actually say stop. Our start codon does not. All right. So that's why you need to remember the start codon, because it's not listed as a start codon. All right. So now what I want you to do is, this is our mRNA strand from our DNA strand earlier. And I want you to write your anticodons. So what's pairing up with it? So I'll do the first one for you. So G, G, and C. And then I want you to find the amino acid that goes with this. And you're always reading the mRNA strand codon to get your amino acid. All right, so don't use your tRNA, because that'll mess you up. You want to use your mRNA codons in that wheel to get the amino acids. Because remember, tRNA is an anticodon. It's just the thing that pairs up with it. We're actually using our mRNA strand to get the amino acid. So go ahead and do that, and then that is it for this section on DNA transcription, translation, and replication.